Hey everyone, just going to go through these exam problems here. For number one, it's talking about an autosomal dominant polycystic kidney disease, or ADPKD, and it affects 1 in 750 individuals. So since it's an autosomal dominant, that means that is the portion, that's the 749 out of 750 would have the disease. So the frequency of the non-disease allele, as it asks in part A, will be your Q. So in order to solve for that, you'll do the square root of 1 over 750, and that'll give you 0.0365. For part B, it asks about the frequency of those affected with it, and so you just take, to find P, you take 1 minus the 0.0365 that we just got in part A, and that is equal to 0.9635. Now it asks what the frequency of heterozygous individuals in the population is. To find a heterozygous population, you have to solve 2PQ. And so you'll do 2 times 0.0365 times 0.9635, and that should equal 0.0703. Now it's saying out of 1 million people for part D, how many heterozygotes are expected? So you take that million times the frequency that we just got, which is 0.0703, and that will give you 70,335 people that will be heterozygous. Moving on to number 2, looking at that figure that it shows of the DNA, it says the letter W above indicates the phosphate group, is what I found. The letter X indicates the ribose sugar. The letter Y indicates a T group, or thymine. It is also a nitrogenous base. And the letter Z is pointing straight to a nitrogenous base. If we look at the shape, it could be considered a G or a guanine, but I think it's just talking about the nitrogenous base. And then looking for part B, it says indicate the 5' end and the 3' end of each DNA strand on the image. So if you're looking at the image, the left side of the strand, the left top will be the 5', the left bottom will be 3', and then on the right side, the right top will be 3', and the right bottom will be 5'. All right. And then it says to indicate the portion of the molecule that is considered to be the backbone. I said on there that the backbone is considered to be the pentose ribose sugars. On the diagram, it would be the line of pentagons that you see on either side. That would be the backbone. And then part D, it says to indicate the portion of the molecule that gives DNA a net negative electrical charge. And that would be all the black circles that you see on the diagram. That's the phosphate groups give a net negative charge to DNA. For number 3, this is talking about the fruit fly Drosophila having either kidney bean shaped eyes or normal, and then ebony body color or normal, and then either cardinal eye color or normal. All right. So first one, part A, it says to determine the allele arrangement in the heterozygous parent. So how we do that is we look for the two phenotypes that are on there that have the most progeny. So in this case, there's one that has a 1,773 kidney cardinal wild type, and then there's 1,761 that have ebony wild type. So we can see from this that the allele arrangement in the heterozygous parent will be K, CD, and then E star over K star, CD star, E. And that should be the parent's allele arrangement in that case. And the next one asks about the gene order of the 3 linked genes. You have to figure out what different possibilities there are based on what you found in part A. So I'll just say the top line that are different possibilities. There's K, C, D, E star, or there's CD KE star, or there's KE star CD. You can have one of those three. There's no other possibilities that you can have. How you find which one it is you find which double cross would produce one of those possibilities that we just listed. And the way you do that is you look at the ones that have the least amount of progeny. And so that would be the ones that have 8 progeny and 6 progeny. The 8 progeny is EK CD star. Then the 6 progeny is E star K star CD. And so looking at that, you find that the K is the one that would have to switch in the double cross in order to produce those results. So you know that the gene order of the 3 linked genes is CD, K, E star. And that's how it is. For part 3, it says to make a linkage map and show the distances between the genes. So first you have to calculate the distances. So first I did the distance between CD and K. You use a single cross to change what you're looking for, and you compare that to the double crosses as well. So you have the 6 on there, the 8 from the double crosses, and then you're looking for, in the progeny that it listed, a progeny that has CD star, K, E star. And so looking for that, I found that there was 97 progeny with that. And then the other one was CD, K star, E, and there was 89 with that progeny. So you add up those 4 numbers, 6 plus 8 plus 97 plus 89. And you divide that by the total number of progeny that there were, which was 4,000, and you multiply that by 100. And that would give you 5 map units between CD and K. Now we need to calculate between K and E, and now you're going to do a monocross with the E star. So we have the 6 and 8 on there again, and then you're looking for CD, K, E, or all the recessive gene, which was 138 progeny. And then the other one is all the wild types, CD star, K star, E star, and that was 128 progeny. So you add up those 4 numbers again. So you have 6 plus 8 plus 138 plus 128, all divided by 4,000 times 100 is equal to 7 map units between K and E. So when you make the map, you'll have CD 5 map units, K 7 map units, E star. All right. And now for calculating the interference, you take your 2 values of map units and you divide each by 100, and you multiply those 2 products together. So you have 7 over 100 times 5 over 100, and that should give you 0.035. All right. For number 4, this is talking about the Chediak-Higashi syndrome. It says that it is an autosomal recessive, so that means that it's going to be the minority will be those that have the syndrome. And it says that 1 in 32,000 individuals are affected. So first it's asking about the frequency of the recessive D allele in the population. And so what you do is you take the square root of 1 over 32,000, because that's the recessive gene, and that should give you 0.0056. And now you find the frequency of the dominant D allele in this population. And so you take 1 minus what you got for the frequency of the recessive D, which is 1 minus 0.0056, and that gives you 0.9944. Next, for part C, it asks for the frequency of the dominant homozygous genotype in the population, and that is p squared. And part B gave us p, so you take that number, which is 0.9944, and you square that, and that should give you 0.9889. And now we're trying to find the frequency in part D for the heterozygous genotype in the population. Heterozygous is 2pq according to the Hardy-Weinberg equation, and so you take 2 times p, which was 0.9944, times q, which is 0.0056, and that multiplied together gives you 0.0111. And that's everything. Hope it was pretty straightforward and that it was easy to understand. Thanks for watching and enjoy.